Hello, everybody. Welcome to this AACI Cards webinar on statistical approaches to bridging gaps in cancer area data. My name is Tesla Dubois. I'm a senior clinical data scientist at the Sidney Kimmel Comprehensive Cancer Center at Jefferson. And we have two wonderful guests here with us today who will be discussing statistical approaches to estimating missing or unknown values in two important areas of catchment area research, clinical trials, and rates of cancer incidence. So I'll be introducing both of the speakers now at the beginning, but we are going to hold questions to the end. And I'd appreciate it if you go ahead and put your questions into the Q&A rather than the chat. We can use the chat for discussion, but we'll make sure to address all of the questions in the Q&A after both of the presenters. So our first presenter today will be Dinesh Mudaranthakam, and Dinesh is an assistant professor in the Department of Biostats and Data Science, a director of Research Information Technology, and co-director of Biostats and Informatics Shared Resource at the KU Cancer Center, and he will be discussing accrual prediction algorithms. And then second, we'll have Jacob Oleson. Jacob is at the University of Iowa, where he is a professor in the Department of Biostatistics, the director of graduate studies, and a director of statistics analytics core at the Institute for Public Health Practice, Research, and Policy. And Jacob will be talking about developing cancer epidemiology maps. So again, thank you for attending this webinar. Thank you to the AACI organizers for holding this great webinar. And I'm going to turn it over to Dinesh, who will share his slides. Again, please put your questions in the Q&A. Thanks.
Thank you, Tesla. Can you just confirm if you can see my slides?
Give it a second. It says yep, we can see him.
Okay, thank you. Good afternoon, everybody. My name is Dinesh, and most of you all, I hope you know me from our recent cards conference. So I'm here to talk about the accrual prediction app that our team has developed and where we are currently with disseminating the algorithm and how you could utilize the algorithm. So before I dive deeper into the algorithm itself, I just wanted to set the ground by giving you all an introduction to our Kansas Cancer Center catchment area. So our catchment area spans across 123 counties, so all the 105 counties within the state of Kansas, but also the adjoining counties from the state of Missouri. So the star here highlights where our KU Cancer Center is. So geographically, we are located right on the state line, like right beside the state line. So this has an advantage, the advantage being to able to serve by state catchment area, but also there are challenges where we have to make sure we bring the data from both registries and multiple different sources and be able to synchronize all this data. So 93 counties among these 123 counties are deemed as rural based upon the RUCC codes. As you can see, the darker red here is urban, and anything in blue or white is all rural area. So we serve a large rural population. So we are a medium cancer center where we have numerous clinical trials that are ongoing. So on an average at any given time point, we have 500 to 600 active studies. And when I say active, either they are actively enrolling or in follow-up stage. So if I have to look at specifically just active enrolling, then on an average, we have 180 to 200 studies. And we partner with our Masonic Cancer Alliance, which is our outreach arm. So they help us take these clinical trials to the doorsteps of our rural participants. They partner with a lot of the sites that are in Hayes, Kansas, or Wichita, or Topeka. So, that's our primary outreach arm. So they make sure they have a constant dialogue going with all these community sites and make sure that whenever we open a clinical trial at the mothership, how can we also onboard these community sites onto the clinical trial? So, majority of these 180 to 200 studies are also open at most of our community sites with the help of Masonic Cancer Alliance. So, given there are so many studies, and as we all know, one of the primary measure for the success to measure the success of the study is the accrual. And how can we easily track accrual across all these studies and ensure that resources are allocated properly? And how can a PRMC committee ensure that while they're monitoring yearly monitoring each of these study, that each study is meeting their accrual goal, or if a study is struggling, how could they give suggestions? So in order to do that, what our team has done in 2016 has come up with the accrual prediction app. One of our PhD students, Jin Hao, has developed this accrual prediction app as part of their dissertation work, and this was published in 2019. So what we are trying to do here is all the information within the clinical trials office. When I say all the information, the basic clinical trial information, such as the PI name, the study title, the disease, as well as the sample size, and when did the study start. All this key information is documented in a standard fashion within the clinical trial management system. And this then is on a daily basis is exported or connected through an API, and then we process using an accrual package. And this is the secret sauce where it processes this data on daily basis, and then it generates the prediction for each study. So the accrual R package is also available through CRAN. But what it does is it uses a Bayesian adaptive, it's a Bayesian design where it consumes the data from the study on daily basis and uses the accrual number as of a given day as a prior, and then generates the uh, date when a study could accrue the overall sample size. So I'll go into more details in the demo here. But in 2024, we have partnered with Inspirata to start disseminating this to um, major cancer centers through their partnership. So they have a robust end-to-end platform where you can build trials within their trial builder platform, conduct feasibility, trying to assess if you have enough patient population across your catchment area, and then how to prioritize your cancers within your catchment area. And there are two modules in their platform that they're collaborating with us. One is this accrual prediction app, which I'm going to show you. And then we're also working on the trial performance module, which we are currently working through this partnership. So here is how the accrual prediction app looks like. So here, if you look at it, the column sponsor ID, study number, the disease working group, the title, principal investigator. If you have a statistician, we often list our statisticians here so that the statisticians can just see by typing their name in the search, they can see which of the studies they are responsible to conduct, interim analysis or, the final analysis, or for any DSMB reports that they might be responsible for. So they can just, keep a tap on their studies that they are responsible as a statistician. And then sample size, expected duration, steady start date, enroll. Enrolled is nothing but number of patients who are enrolled as of a given date. So this refreshes on daily basis. So the numbers I would be seeing 12 for this first study would be as of yesterday, March 24th, 2025. Um, and this is all simulated data. So again, there is nothing confidential here. So what the algorithm does is it takes the sample size, the expected duration, the start date, and number of patients who are enrolled as of the given date, and then it calculates or estimates the estimated end date and a lower bound and an upper bound. So this is a confidence interval, 95% confidence interval. So we also calculate the probability of being late, and I'll show you in the next slide how this percentage is being assigned. But a little more details on the screen. We also added like a health check. So anything that is highlighted with a green colored checkbox or anything that is highlighted in yellow, our PRMC committee is not too worried. But anything that has a red means they're not meeting their accrual goals. So how can they improvise? And this is where either they work with the informatics team to figure out how can they leverage the electronic medical record, or can they work with marketing team to advertise their study. So this is where, again, reassessing what could be done in order to invest the resources in a right way to ensure the study continues to enroll more patients and achieves success. So here is where the graphs, you could see some graphs here. So just to explain, um, and how do they get to this graphs? If they click on the study number itself, for each specific study, these in specific graphs are displayed. The top graph is showing the number of patients that could be accrued in a fixed time. The bottom graph is a flip. So in order to achieve the fixed number of sample, what is the time it takes? But you can look at this little box here, which says, so far this study, and this white line here is the actual um, count of how many patients we have accrued. And this red line here is the duration, which is the 24 months duration. So for this study, they're anticipating to enroll 8 patients, and they have 24 months to do this. And so far, they have enrolled only 2 patients, and they have already lapsed little more than 5 months. So in order to reach the 8 patients, we are saying it would take 22.3 months. If they can, expedite it, this can be done in 10 months, which is the lower bound on the 95% confidence interval, or it could go all the way to like 60 months. So again, this kind of information, when provided to the PI as well as the PRMC committee, it allows them to make a decision whether to keep the study open, or do we want to reassess on how to move forward with the study. And typically our statisticians call this is like the tornado. When you look at the weather reports, whenever there is a tornado prediction, this is the space, and the white line is the eye. So it is similar to that concept here. So this is another example here. So if this study is requiring 80 as a sample size, and time to completion is 24 months, and participants recruited to date is 64, and so far it's already 25 months. So this is a little beyond the fixed 24 months time point. So at that level, we don't give a prediction for how many they're going to recruit in 24 months, because this is already past the 24 months timeline. So here you can see for 80 patients, we are suggesting at this rate, it's going to take close to 31 months, and soonest we can get there is 28.3 months. So again, the reason um, uh, we say the soonest and this is how it is going to take is, again, it's adapting. So if we enroll 2 patients today, then this would be 66 patients, and all these months and the prediction will change and update itself tomorrow. So all of this is automated behind the scenes with connection to our CTMS system. So here is the other view that we have developed for Accrual app, where this is not a tabular view. The earlier view was a tabular view. This is more like individual study level view. Even here we have the color coding going, where we have red and green and a yellow. And at times people ask like, okay, our study is lagging and we are struggling to recruit, then what do we do? So for that purpose, at the study level, they can simply click on find more patients, and using the inclusion exclusion criteria, we could try to retrieve participants from the EMR and figure out who are the participants who may be qualified to be on the study. And again, below is how you can see the study, and here are some numbers where there are eligible participants, it will show you. And the inclusion exclusion criteria here is something that you can flexibly add or remove depending upon again, individual study and the PI's requirement. So now I'll quickly go through a demo of the application. I hope you can still see it.
Dinesh, is there sound that goes with this demo?
Oh yeah, you're not able to.
Sorry, we're not hearing it now.
Okay, I'll just give a voiceover. So what we are showing here is the platform where we have features that are built in, where we can search based upon any of these columns or sort any of these based upon any of these columns. And then there are two different views, like how I was showing. One was a grid view, and another view is the individual view. And again, the final thing, I've already gone through these plots. The final thing is right here, we can directly click on find more patients for studies that are struggling. And um, using this feature and functionality, you can find additional participants who might be qualified to be on the study. and the screening team or the coordinators can directly start screening the participants. So that's what we're seeing here on the screen. Sorry about that, Tesla. Thank you for letting me know.
Yep, no problem. Thank you.
And I want to just thank team and Inspirata's team. And I want to remind people that we are trying to write a white paper based upon our design. So if you're interested in testing it out, or if you're interested in providing your feedback about this trial accrual prediction application, please reach out to me, or you can also complete the survey. This is the Microsoft survey, so you can just complete the survey. Thank you.
Thank you so much, Dinesh. Please go ahead and dump all your questions for Dinesh into the Q&A. And in the meantime, as those accumulate, we will hear from Jacob Oleson.
There we go. Well, hopefully you're seeing my screen at this point. Thank you for having me on here. I'm happy to be here to talk about this. You've probably not seen me before. Talking about developing cancer epidemiology map to detect hotspots for small geographic areas. But hopefully you've seen my colleague Sarah Nash, who has presented some of our work. But she has asked me to come on here and present more of the statistical details behind the scenes of what we're doing in our project. So I'm going to start at the end. This is our product, CAMSA, which shows the cancer rates in small geographic areas. So I want to talk a little bit about the behind the scenes of getting to this point. And I think in order to understand why choices we made along the way to present some of the things that we do in the way that we do, it's helpful to go through historically how we got to this point. And I'll add to that this site finally became live about 2 hours ago. So this is a screenshot of it, but the live site is up, and I probably should have it on this page here, but I have the actual link at the end of this talk. So going through historically where we're at, the Iowa Cancer Registry came to me more than 10 years ago, asking about presenting cancer risk at across the state in small geographic areas. So we talk quite a bit about what do we mean by small geographic areas. Is it zip codes? Is it census tract? Is it county? Is it a smooth spatial surface? And coming from a rural area myself, I wanted to be able to have the general public go into our tool, our map, and be able to find where they live specifically. Which then we want to go to a small geographic region as we could in order to do that. And I think most people don't know specifically what census tract they live in, but they do know what zip code they live in. So that led us to trying to develop this at the zip code level because it was targeted towards a broader audience and not a research audience. And that leads us to rural populations because, as you might imagine, just like you saw with Kansas, Iowa is also a fairly rural state. So as we're looking at cases in these small geographic regions, we're going to be dealing with small counts. We're going to be dealing with zeros. So we need to come up with a way in order to mask that information in some way. And another decision that we've made along the way, and you'll see more of this as we go through, is we want to present something that's as close to the raw data as possible, but we can't present the raw data because our counts are too small. So they have to be model estimates. So what that means from a statistical modeling perspective is we want our models to be adjusted for as few things as possible while still masking um, individual identifiability. So those are some of the pieces that we had in mind as we were going through developing what it is that we developed. So our initial start, and this was 10 years ago, the very first draft that we had creating maps for using statistical methods to quantify cancer risk in small geographic regions. And at that point, you see the 3 time points, 2004 through 2007, 2008 through 2011, 2012 through 2015, 3 different 4-year time periods, which led us to these particular maps, which until 2 hours ago were on this website. And so the thing to point out with this is, yes, as you hover over, it points out exactly what the town is, what the zip code or zip code tabulation area is that we're looking at. But these are not on we're not on a relative risk scale. They're not on a age-adjusted rate scale. They're on a probability scale. So, in order to get to that probability scale, so that's what I'll walk through with these statistical methods. We have a zip code tabulation area. The data that we have age, sex, year of diagnosis, and death stage for every cancer in Iowa over that time period. And we had decided to focus on these 8 specific cancer sites for the 935 ZICTAs in Iowa. So small data sets. For colorectal cancer, one of the more common cancer sites in our state, we still have most of the ZICTAs, 750 roughly of the 935, are going to have fewer than 10 cases where we wouldn't be able to show the raw data. And as we are looking at standardized rates in some way, back then, 2010 fell in the middle of that. So 2010 was the reference population distribution that we were using. We, for that, built a hierarchical Bayesian Poisson model, a standard disease mapping model for count data. And we looked at a lot of different variations of models. I have 3 of them up here. We ended up going with the 3rd one that's on here. And what these letters are referencing is we don't have any variables that we're adjusting for. The intercept beta naught is just an overall state average. Z represents our spatial effect, how similar are neighboring regions. T represents our time effect. Each region is going to have a similar rate from one time point to the next. So T represents that correlation across time. And the last term, the delta T, allows each zip code to have a different pattern over time. It's an interaction term between space and time. So that was our model. We have space and time, but no other covariates that we're adjusting for. And this is to estimate the relative risk. Our spatial effect and time effect very generally are written in here. The spatial effect is what's called a conditional autoregressive model. Technically, it's the intrinsic conditional autoregressive model. We take each of the 935 ZICTAs and which ones share a border of any, no matter how much border you share, just do you share a border or not? That's a yes or a no, 1 or 0. So neighboring borders are going to have similar rates to each other. And then over time, we have an autoregressive model of order 1 so that each time point is correlated to its previous time point. The result of that, this would be the relative risk where the state average should be 1. And you can see there's not, these maps are not all that colorful at the relative risk scale. So we wanted to take the relative risk and then go further away from the data than even this is. So the way that we did that was to construct a risk probability. What is the probability that your zika has a relative risk that's above the state average? And because we're doing a Bayesian model through a Markov chain Monte Carlo process, we can take a look at that whole distribution of values and create a probability that each one of these zip codes or zictas has a relative risk above the state average. It's a Bayesian exceedance probability. Shorthand, we call that a risk probability. So on the top, we see the relative risk, and then a direct comparison to the risk probability is on the bottom, and then that's what we reported on the website, the risk probability. And it is not possible to go from those numbers that are in that bottom map and go back to what would be the raw count. There are too many steps, too many statistical layers in the middle to do that connection. So that's how we protect the privacy, and we give a more meaningful map on the risk probability. So as a statistician, I like the probability measure. Probabilities make sense, but I forgot once this goes out in the general public and our epidemiologists go about sharing these probability maps. It's like rainfall. When you say there's an 80% chance it's going to rain tomorrow, well, it's either going to rain or it's not going to rain. It's 100% or 0%. Anything in the middle is 50-50. So most people only think there are three probabilities that matter. So we have this whole range of probabilities, and people have had a hard time understanding where these come from. They wanted age-adjusted rates. So we have finishing up an informatics technology for cancer research grant, where we took all of this and converted it into an age-adjusted rate. As we're doing that, there are going to be some problems. The proportion of zeros, if it was bad overall, now we're going to stratify by age groups and look at how many cases there are in specific age groups. In colorectal cancer, there were 4% zeros before, but by age group, now we have 63% zeros. So zeros are a much bigger problem at this point, leading us to create an additional model for estimating age-adjusted rates for low prevalence diseases over space and time through a hurdle model. We construct a crude rate for each age group is effectively how the model works. We still have a Bayesian model, it's still a Bayesian Poisson model for the most part. And it works per age group. And then once you have the age group specific estimates, then you combine them together through the direct standardization weighting, which again, we're going back to the Census Bureau weights for that. We did construct this as a Bayesian hierarchical hurdle model, which accounts for the excess zeros by saying if there are counts 1 or above, it follows a Poisson distribution, which is the bottom part of this. If it's a 0, the first question is it a 0 or not? And that will follow a logistic or binary form. And if it's not a 0, then it follows a Poisson form. And we set up a model for the logistic, the Bernoulli regression part of it. It has, again, we're not adjusting for any covariates. We do have population size to adjust for of that region. And we have age group. This gives us our age group specific estimates. This is a spatial effect. So again, we put a conditional autoregressive to allow neighboring zictas to be related per age group. And we allow neighboring time points, one time point apart, to be related. In addition to the Bernoulli model for the zeros, then we have another model on the Poisson, a population size, age group, a spatial effect, a time effect and additional random error. This looks very similar to the model before. And as a Bayesian model, we put in additional priors, and we had a lot of discussion on how to select our priors. And for the most part, they are proper priors that are very vague. So like the beta coefficients would have a mean of 0 and a variance that is very, very large. And finally, in order to get an age-adjusted rate, we have to combine all that information back in together. So that paper that I referenced a few slides ago walks through this formula for how to bring those estimates back together to create an age-adjusted rate. And so this is a rate per age group, which is K, and then we weight those back together to get an age-adjusted rate per zikta per time point. And in addition to having age-adjusted rates, we also compute those risk probabilities as well. So the Bayesian exceedance probabilities are computed the same way as we did before. We now have an R package that runs this. So for any state that wants to work with us, then we have an R package to run the data. We have code that works in OpenBugs, INLA, and Nimble for those familiar with Bayesian models. Our R package is built for Nimble. And these we have crude rates by age group, we have age-adjusted rates, we have risk probabilities, and we have whatever specific cancers we put into it, and then create our maps. So as we were doing this, we had the hurdle model and the Poisson. So I need to step back a little bit and compare these. And if you stare at these two maps long enough, these are at the county level for Minnesota, Wisconsin, and Iowa. Looking at run once with a hurdle model, and then once ignoring the excess zeros and looking at just the Poisson. And there's almost no difference between these two maps. If you stare at them long enough, you'll find a few counties where there's a slight change, but there are very few of them. And we've investigated this further. We still have some more investigation to do. But as the population sizes get really tiny, the hurdle model does start to break down. We need to fix that. Whereas the Poisson model works no matter what our population size is. And when we get down to ziktas at the age group, that really matters. So for now, all of our models are strictly Poisson, and we do not do a zero inflated adjustment of any kind. But we do have verification here that it doesn't really impact our results, although we do want to fix that going forward. So the remaining pieces, that's most of the statistical piece behind what we have. And then is what does our tool look like? Well, we have an interactive web-based application. There are 8 cancers that we have in our tool for Iowa. We have geographic units. You could select either a zikta or county. You can use either one. We stratify what we're going to put on the website is sex and year. We can run it by race, but for our model for now, we've removed that. And we have age-adjusted rate, risk probability, and a population estimate. And we can also highlight hotspots. So this is the map I showed you earlier. This is the resulting product. And as you hover over the zip codes over here, it'll populate what the zikta is, what the city is, the age-adjusted rate, a standard deviation, because these are model estimates, a risk probability, and a density of that region. And on the left are all the choices that you can make of what you want to look at. And there are a few other choices over here on the right. Well, those are the choices on the left. This is a description of what the numbers mean. We've done panels to help get the wording here so that it's more meaningful to a broader audience. And as I said, there's a description of what is being shown. And then we can switch that over to a risk probability instead of an age-adjusted rate. A lot of people like the age-adjusted rate. They're more familiar with it. As a statistician, I like the risk probability measure better. we can filter if we really want to find hotspots, since this is a Bayesian probability-based estimate, which ones have a greater than a 95% probability of being above the state average, and it will immediately highlight those areas. And below it will be a table that showcases all of those regions. I said that over on the right, there are other things you can add. So behind the scenes, we can add where the counties are. We can add roads and borders. We can add city labels. We can add other information to make it easier to identify where these specific regions are. And then you can zoom in and out of those. So that was a quick rundown of what our modeling is. We have a large team of biostatisticians, epidemiologists, geographers, and a patient advocate to help us put something together that could be meaningful to a broader audience. You can see the NCI grant that has helped us put this together for the past 3 years. And there is the website if you want to go check out our map.
This is great. Thank you, Jacob. Awesome job. So I don't think anybody else can participate in giving our presenters a round of applause, but here you go from the 110 people that we have on the webinar right now. So we can go ahead and field some questions. Both of our speakers can remain on camera, and we have a few questions for each of you here. Dinesh, one of the questions that you have here in the chat is about looking, and I think you answered it a bit in here too, but your modeling is looking at the prior accruals for clinical trials in order to kind of estimate when you'll have sufficient accrual and whether or not you'll reach sufficient accrual. How far back is that model looking, and how many kind of time points do you need for that prediction to be able to be made?
So it is considering just the study characteristics. It is not using a different study or a prior studies information that could be added if we want to. But there is also a package that is out there that our researchers use it for the feasibility during the feasibility analysis, where you haven't accrued any patients yet, which will be the number of subjects would be 0, and then time elapsed will be 0. So you can plug those in to the feasibility platform, and then you can try both. You can try with trying to get at how many subjects you're going to get if you have a fixed time, or you could also try the other way around where you have a little bit flexibility with your time, but you want to get 100 participants because you want to reach certain effect size and power. Then you can see how long is it going to take.
Okay, so in that case, can study teams essentially estimate the months for the study at the feasibility stage?
Yep.
Awesome. All right. And it sounds like there are a couple different use cases or benefits to using this accrual prediction application then.
Absolutely.
Is your team planning to enhance the prediction application on top
Yeah,
of what you showed us
so
today?
yeah, great question. So what we are trying to do is we are also working with All of Us program, where they look at a UBR, underrepresented population. So we have different criteria there. They want to ensure uh, the amount of participants we enroll under that program as, rural or Hispanic or, a particular race. So each of those centers or HPOs, like how they call, have a specific criteria written into their contract, and they have a set threshold. So KUMC has obtained the contract to be participant in the All of Us program, and our criteria is we need to achieve at least 60% of our UBR, which means enrolling the minority population or underrepresented population. So what we are trying to do is we are trying to add or enhance our algorithm to include and be able to predict based upon how many patients they have enrolled already, to show like within the next year or within by the end of the study what percentage of our sample size is going to be underrepresented population. So that is a new enhancement that one of my PhD student Kastub is currently working on, and we hope to roll it out here in the next year.
Got it. And a question from Chris McNair in the chat for you, Dinesh. Are there data sources that are specifically available within the Inspirata suite, or are most of these data sources that you're using institutional?
So the way how we have partnered with them is mostly institutional, but they do have other data sources that sites could leverage. But I think I can reach out to Chris and give him more details. I don't want to speak on behalf of Inspirata because that would be
Yeah.
inappropriate.
Got it. All right, jumping over to you, Jacob. So I thought your modeling was really exciting. I'm doing a little bit of Bayesian stuff, but I haven't seen this two-step piece before, so that was pretty neat. What do you think the biggest barrier would be for an institution to reproduce this with their own catchment area? Again, coming from Chris.
So I didn't mention this in the talk, but we're working with the state of Kentucky and the state of New Mexico right now. With Kentucky, we have shared our R package with them. They're running the model on their data because they didn't want to put, or were not able to put a data use agreement together with us. And so they'll run it, and then they can share the estimates with us. We can recreate the maps for them. With New Mexico, they were able to put a data use agreement together to share their data with us. So they share their data with us. We run the model, create the maps, and so we have both of those ready. And we've submitted a new proposal to extend our work into other states, and it's being reviewed next month, actually.
Okay, that's exciting. And you have, I heard a lot in your talk, this concern about the population size, and I'm curious, there's a question here about why race wasn't included. Is that related to, just having too many zeros within a census tract?
It's partially related to that. It's more of an interpretation question that we were not ready to put that out there yet. And that's because of the way the statistical model works. So as a specific example in Iowa, let's say I want to do estimates of the Black population for cancers. A vast majority of the zip codes will have a zero population and a zero case count. And so then our estimates are not going to be zero. Our estimates are actually the state average, because the way the model is built is it gives it the intercept, which is the state average, and then the spatial and time effects say, is there enough information in your ZICTA to pull it away from the state average? And if there's not enough data there to pull it away, we will be showcasing a state average estimate, which we know is wrong, but there's no evidence to pull it away from that from a statistical standpoint. And so trying to describe that on our website to a large, broad audience, we're not ready to do that yet. So we're trying to figure out how to communicate that, or if we should change up how the model works so that it is weighted down towards zero because the population is zero there. So that's why we've removed race for now. The model works.
How much change are you seeing using your time sensitive models? I'm wondering if kind of grouping buckets of time could help with that stability piece as well.
Yeah, the more data that is in a region, the more stable it's going to be. The less data that's in there, the more the spatial smoothing and the time smoothing is going to impact that estimate. So for these smaller areas, there can be quite a bit of difference between the raw data estimate and the statistical model estimate. And we've looked at that. Things follow patterns like we expect them to. But if we're going to show small area regions, that's just what we've decided we're willing to live with.
Right, right. So you have a question in here from Johnny Rose from Cleveland. Do you feel comfortable making comparisons to other age-adjusted rates for other populations standardized to the U.S. standard population when using the modeled ZCTA level rates?
Yes, hi, Johnny. Yes, I do feel comfortable doing that. In fact, we are doing that. But also, there are the caveats of then we're using an estimate, comparing against an estimate, throwing away all the variability that's involved with both of those. And so there's a certain level of trust you have to put into that estimate then, because we know we're throwing away variability. But yeah, but the short answer is yes, we are doing that. Just understanding the caveats that go with it.
And then another question related to the ZCTAs about where you're getting cancer incidence and mortality data. I believe you're working with registry data, is that correct?
We are working with registry data, yes. And so we have all of
Okay.
the cases for the.
And then go ahead. Somebody. Oh, no, okay. And then so there's a question about why you went to improving the hurdle model when the Poisson model is successful even at lower sample sizes. And could you not use a zero inflation Poisson model instead?
So why use the hurdle model? I'll answer that one first. Because we're comfortable with the Poisson model. We think it gives good estimates, but it'd be a great PhD dissertation to develop the hurdle. So that's part of the reason to do that. The zero inflated, we do have a zero inflated model that we could use. That the reason not to do that is more of a theoretical decision on our point. The purpose of a zero inflated is that you see a zero due to random sampling. And if you were to take a different random sample, you might actually observe the cases that were there. So then it would no longer be a zero. That's why you have too many zeros. In our
Hmm.
case, it is registry data, so we have the entire population. If we did this again, it'd be the exact same data. And so it's not a random chance that we're observing zeros. We're just observing a lot of zeros. And the reason the Poisson still works is that the distribution of the Poisson still works pretty well. If we look at what should the distribution look like for a particular mean, it still follows pretty well. So the hurdle model isn't necessarily needed, but it's an interesting theoretical statistical question.
Right. Okay. Um, and then one more question for you, I think so far, from Jason Lau. Thanks for the nice talk. I wonder in general for hotspot analysis, how is your smooth risk rate stratification versus other clustering methods such as SAT-SCAN or JEDIS board?
Great question. Actually, the back when I was asked to do this over 10 years ago, it was a SAT scan estimate that they had in there. They had a continuous spatial grid. And then I came in and we switched over to this format. And one of the primary reasons was that I did not, and this was discussion with our cancer registry, of using a continuous spatial grid where it's harder to determine where individuals live over a continuous space that the SAT scan would give, at least at that time, versus this kind of modeling. And then because I'm a modeler, statistical modeler, the Bayesian part of it gives us better probability estimates than the SAT scan does. And so I like that probability aspect that comes out of it.
Okay. And then from Dr. Blackman in the chat here, I think specifically to your application, the CAMSA application, when comparing to the SAT-SCAN application, what kind of improvements or, cell would you give to an institution kind of comparing these two applications for analysis?
Well, I would have to look at that specifically. I can speak generally, but I haven't looked at that to know for sure.
Okay.
Generally, it's we have very specific geographic regions. It is we do not have any requirement for sample size, population
Mm.
size for a region in order to put this together. And then we get age-adjusted rate that does adjust for the population size of each of the individual regions. And we have a Bayesian probability behind it that only statisticians like me probably care about. But I think that that's one of the benefits.
Right. You're there's a lot of interest here. The questions keep coming, so I'm going to
Sorry,
keep you
Dinesh.
talking here. So did you consider other count data models besides Poisson? For example, the generalized Poisson model.
Yeah, so the a hit against a general Poisson model is that you force the mean and variance to be equal. And because this is a Bayesian model, we can make up our own rules along the way. And you can see in the models that there's an additional error term in our Poisson regression models. And that
Hmm.
additional error term is the overdispersion that you would see in either a negative binomial or a generalized Poisson model. So effectively, that's what we're running.
Okay. All right. All right. Last question here, I think. Can you identify cold spots in addition to hot spots in your modeling?
Yes, we can. It's not on our tool right now to identify. So we can identify 90% and above, 95% and above, but can we identify 5% and below, 10% and
Mhm.
below? Our models do. We don't have it put up together on the CAMSA application yet, but we have that information available.
Okay. And then how long will you be updating the website? How
Uh,
often?
we
Sorry.
we plan on updating it every year as we get new data from the cancer registry. Then every year we can update it.
Okay. All right. Wonderful. Well, give it a couple seconds here, but I think that we have cleared the chat. Thank you both so much, Dinesh and Jacob, for your presentations. This was really engaging, and I know I have plenty to think about coming away from this. And thank you again to our hosts at AACI, Kate and Haley especially, for getting us prepared for the webinar today. Thank you so much to everybody who came and stuck through to the end. We had a great turnout, and I wish you all a wonderful rest of your week. Take care, all. Bye bye.